Hello, everyone. Welcome to our webcast today. Thank you so much for joining us. I'm Cheryl Rogers, the Director of Marketing at Golden Helix, and today it's my pleasure to introduce to you Dr. Jeffrey Rosenfeld of the Rutgers Cancer Institute of New Jersey. Jeff will discuss today with us a workflow currently in place at the CINJ for a 50-gene panel that identifies mutations and hotspots. Jeff, I will pass the mic to you.
Okay, thank you, Cheryl. I really appreciate everybody tuning in. I've done a lot with Golden Helix, and glad to share with you.
Unmuted.
So,
Muted.
just a quick bit about me. I'm assistant professor of pathology and laboratory medicine at the Rutgers Robert Wood Johnson Medical School. Basically, as anybody in academia knows, you have to be in a department, so they put all of genomics in pathology here at Rutgers. I supervise the computational aspects of the Personalized Medicine Initiative at Rutgers Cancer Institute. This is where we're doing sequencing of tumors. Also, I'll discuss we're doing some work with Foundation. We're doing a lot more on trying to bring things in-house and do sequencing of tumors in our own lab. We're the only NCI-designated cancer center in New Jersey. There's one of 35 nationwide. And I've been working with Golden Helix for a long time, since 2009. Did a lot of work with SVS, and now I've been working a lot on VarSeq. So cancer is traditionally treated with chemotherapy with all of its harsh effects. And mainly the way people would treat cancer is that they would find a site of the tumor, and based on the site of the tumor, you would treat it. There would be a certain type of chemotherapy for breast, certain things for lung, certain thing for colon, that's the way it would be treated. Now we're realizing that instead of just focusing on the site of the tumor, the mutations are much more important. So what happens is that there's a mutation that causes tumor growth. There's a tumor that's addicted to some mutation that driving its growth. There's mutation, or there's a SNP that's causing that tumor to, and a gene that's causing the tumor to grow, and if you can attack that mutation, you should be able to destroy the tumor. So the first time this happened, that someone figured this out very well, was the case of the BCR-ABL fusion for CML. This was known for quite a while, and there's a drug, Gleevec, that was developed, which attacks this mutation. And there's a very strong therapy for CML. There's also things like BRAF and other mutations that are found in lots of cancers such as melanoma, and it's been found that whether someone has or lacks one of these mutations is a clear indicator of how they should be treated. So this has become a big thing. There's all these slogans, personalized medicine, cancer is personal, know your tumor, my cancer genome. There's all these things that are saying that really cancer is not one disease. Even within lung cancer, there's different mutations, and you have to treat the person's tumor based on their mutations. So the real key to this is finding druggable mutations. There's 70 FDA-approved drugs that are targeted for cancer mutations. A lot of these drugs are Me Too drugs that target the same mutation, so it's not exactly 70 mutations that are targetable. But some of the drugs are, but in many cases, these drugs are being developed. Cases are developed for one type of cancer, and doctors can off-label prescribe it in another type of cancer. So meaning if mutation is normally found in lung cancer, and a patient has breast cancer with the same mutation, the doctor can prescribe that off-label if they think it will work. While the drugs are making their way through clinical trials, there's two ways. There are some drugs that are specifically designed for a certain mutation. The other is that there are mutations where the drug doesn't do well generally in approval, or even more now, the FDA is asking for genetic testing to be done as part of a clinical trial, and then it's found that overall in all patients, this drug didn't do well, but in a subset of patients, a certain mutation or set of mutations, the drug did very well, so that's being used. So here's the procedure. There are two different types of sequencing. There's tumor only or tumor normal. This shows tumor normal, where you take the tumor or do a biopsy, put it on slides. You also take either a buccal swab or blood, or you take adjacent normal tissue. You sequence the two of them together, analyze them, and then you get a list of variants. The variants are sent to the sequencing tumor board, a molecular tumor board, which analyzes all the data. They take all the sequencing mutation data in line with the patient's characteristics, their medical record, and decide what to do with how the patient should be treated. And then that's given to the doctor, and the patient is treated properly based on that. So to determine mutations in a tumor, as mentioned, there's either a tumor only or tumor normal approach. So the tumor normal approach was introduced much earlier. The idea of tumor normal is that you can if you sequence the normal, that'll give you control of what mutations are not important in this tumor. Because those are in the person's germline, they're sitting in all of their cells, those are most likely not responsible for the tumor. There are mutations in germline that can be found in tumor, but that's fairly rare. This is what was initially done. You do this subtraction here, and you get mutations only in the tumor. More recently, it's been found that because of the databases of public data out there, TCGA has 11,000 samples, the International Consortium has 17,000 samples. We're getting a lot of tumors out there, and based on the data from other tumors, you can easily look and say this mutation is likely to be pathogenic or not, and you save the need for sequencing the matched normal. The other reason for the trade-off is that sequencing 2 samples of tumor and normal is going to have double the cost because you have to sequence 2 samples. You could argue that it's not exactly double the cost. Some people sequence the tumor to higher coverage and the matched normal to lesser coverage. Some providers build the cost in where they're only sequencing at a lesser amount, and then it's 1.5 times the cost rather than double the cost. But either way, you're paying extra for the mutations in the germline, and we found it's not necessary, especially when you work in a health system or a hospital. You're trying to figure out how many patients can you sequence with a set budget. It's not worthwhile. So there are 3 ways for hospitals to implement tumor profiling. The first idea is you could send everything to a commercial lab. This means you take the tumor biopsy, you send it to a lab, and then the lab does everything, and basically the lab does billing and everything, and all you get back is a report. I'll show you. It's very easy to implement. All you need to do is send slides, have the pathologist mark and send slides to the company. The problem is that you only get back the report, and you can't necessarily customize the report. You can't add or subtract genes that you would want. It's also hard to get access to the raw files. If you want to do research like we're doing here at a research hospital, you really want BAM files or some type of raw data rather than just the result file. You want to know how many reads there were, what mutations were found, and pull all the data together, and you can't really do that with a lot of commercial labs. So here's a bunch of the commercial labs that are out there. You can see Foundation Medicine is probably one of the leaders. We've done a lot of work with them. There's Personalis, where they sequence, instead of DNA, they also sequence RNA, so people are moving to that also. I could have put another 10 or 20 companies on here. They're starting to take over all different companies that are doing tumor sequencing. Different people have different size panels. Some people do 10 genes, 100 genes, 400 genes, a whole exome. There's a big discussion, which I can get into in questions about how many genes are necessary. Because part of the thing is that if you do more genes, you might just be getting more VUSs. And so if I sequence, instead of sequencing the 400 gene panel, I sequence a whole exome of 20,000 genes, or 50,000, depending on how you count genes, all I'm getting are more VUSs. I don't know what to do with them. It doesn't really help me. So there's a bit of a trade-off, and some people do a larger panel just because it's a marketing thing to say, we have a larger panel, we're better than other people, but it doesn't necessarily mean it works clinically. So after you run the report, you get something back. This happens to be from Foundation Medicine. It shows that these are the mutations that are detected. Here are the FDA-approved therapies, so these therapies are approved for that particular cancer, for that mutation. But let's say these mutations were already, these drugs were already tried, the tumor is resistant, then you can move on to these drugs that are put in other types of therapies, and then you can look at clinical trials. If that doesn't work to try and put somebody onto a trial, that's step. There's also potential toxicity for the different drugs, but that's generally the way it works. Some doctors just get this back and then act on it directly. We happen to have a tumor board that reviews these recommendations to make sure they are accurate and usable. The second approach, which is the polar opposite of sending everything out, is something like the MSK Impact study. So MSK Impact is at Sloan Kettering Cancer Center in New York. What they do is they've done everything in-house. Everything is customized. They have the panel of genes they designed. I think for the pull down, it's either an adjuvant or a Nibelgen capture. But besides that, everything is customized. They customize the number of genes. The software is built by their local developers. It's customizable. The database of mutations and how mutations going from mutation to the clinical actionability and the drugs and the clinical trials, it's all done in-house. This allows for complete control over the data and is very good. The problem is this takes a huge amount of resources, takes a lot of people, a lot of money. You know, all types of people. You need software developers, pathologists, people just annotating papers, all these things in-house. And unless you're a very big health system that has a lot of money, it's going to see a lot of cases. It's very hard to justify and to do this. So then there's the hybrid approach, which is what we end up doing at Rutgers, is where you run the test locally using different commercial parts. So basically, there are a lot of parts I'll explain that you can plug and play to do different things. There's companies that will produce the panels, analysis software you can buy, and there's even different parts of the analysis software I can buy. And here you maintain control of the data. The results you get back are yours. You're controlling how everything is working, where it's going, but you don't have to do the work. You can, also nice is if one of the providers is not good, you can easily swap them out and get another provider, get something new to put in there. And so that's the approach we've done at Rutgers. So in a nutshell, here's what we're doing. We do the Radiance Thunderbolts panel for the DNA enrichment, run on a MySeq. Basic analysis is done in BaseSpace, then we use VarSeq, and then we run it through N-Of-One. So the reason we did this, it's a targeted 50-gene panel. It's designed to be very low cost and a quick turnaround test. So the idea is patients who don't find a mutation that makes sense or that is targetable on this test would reflex to a larger panel. So this is the first step in the first line testing rather than sending everybody to a larger panel. So here's Radiant. This is what the machine looks like. Put DNA in. What it does drop the PCR for 50 top cancer gene hotspots. So the reason droplet PCR is good is because rather than doing just a standard multiplex PCR where you throw in all the pairs of primers together, in that case, with a multiplex PCR, you end up getting enrichment of certain primers over other primers because they outcompete the DNA, and you end up having a very unbalanced mixture of your amplicons. So rather, this is droplet PCR. Every droplet has one pair of primers and a DNA, and so it can target that DNA and just enrich that location. There's a total of 200 amplicons, and so the total of the amplicons are only less than 40 kb of DNA, which is critical to allow us to get very high coverage even with multiplexing of this. But the other thing to remember is that this does not cover full genes or even all exons, only hotspots. So meaning you sequence part of these genes, the patient could have a JAK2 mutation that you wouldn't find because the JAK2 mutation is either on an exon that we're not targeting or even a non-coding SNP, you wouldn't find that doing this. So that's the one drawback. That's why, but as I said, this is a cheap first-line test, and so that's why we're running it. Samples are multiplexed. You can put 8 samples together on a MySeq run, which is very cheap if you know the cost of a MySeq, rather than a HiSeq, where on a HiSeq you could need 2 or 3 per lane, maybe even 1 per lane, depending on the size of your sample, size of your panel, so that makes things much cheaper. The alignment and variant calling are performed in BaseSpace using standard tools. But the key thing is that all we can call on this panel are only SNPs and indels, which is very good for some things where you only want to find SNPs, but there are certain things like in lung cancer you really want to find ALK fusions. And while we have ALK on our panel, we don't detect the fusions because we're only doing the short amplicon sequencing and not the full gene. We also can't call amplifications or deletions, which are critical in a lot of tumors, and so we're not getting that. But really what this is doing is the most highly studied tumor mutations. So rather than doing a panel for 1 or 2 mutations and doing it on a Sanger, this replaces it in a cheaper and faster way for doing this full panel. So another thing that came up that I didn't really realize, and that to do this type of testing, there's a whole lot of IT work that needs to be done. We're CLIA CAP certified and HIPAA compliant shop, so to do all that, data transfer and the data storage are very complicated. Our sequencing lab and our analysis machines are on different networks with a firewall in between. So we need to do a lot of encryption and opening holes in the firewall to transfer the data. The BCR files are encrypted and copied. The way it works is that on the sequencing machine, as soon as the cells are processed and a VCF is produced, it goes into a folder where there's a cron job, and that automatically takes anything in that folder, encrypts it, copies it, and archives it, and sends it to the analysis machines. The other reason for the backup is that since these are clinical records, they need to be saved for a long time, depending on who you ask, 5, 10 years, or even longer. And this is a very critical thing, how to store this data, how to build a machine, because once you build a machine, you don't really want to have to swap out the machine. So you have to spec out how many cases do you think you'll run over the next 10 years, and then find a way to store all of that and keep it. So for the analysis software we're using, there are a few different trade-offs. First thing is commercial versus open source software. Commercial software, you get robust support from professionals. You call somebody or email them and they'll respond to you fairly quickly, which is very good. The problem is, of course, you have to pay for the software. As far as open source software, as anybody who's worked with open source software, it's usually written by a grad student or a postdoc. And then what happens is the grad student graduates, and then, unless it's a really important piece of software, the software kind of dies. You look at GitHub, the program hasn't been updated in 5 years, you're not sure how good it is. And that's the critical thing people have talked about in the field, how to get software to be updated and how to get the life cycle of software to keep going. But that's the trade-off. The other trade-off is graphical versus command line. For a non-clinical person, such as a clinician or even a wet lab technician, they would much prefer a graphical interface where you can point and click and do everything. Whereas a bioinformatician likes to automate jobs and build pipelines. And the more technical people are much more comfortable at a command line than they are with a GUI, because a GUI you have to point and click everything rather than being able to do things in a repeatable manner automatically. Final thing, which is even more critical, is the idea of a pay per sample versus a license model. So pay per sample is what a lot of companies do, where every time you want to run a sample, you have to pay either hundreds of thousands of dollars to do the analysis. And this, depending on how you're doing things, if you're billing and getting reimbursed, then you can just put this in as a line item on the bill for the analysis of the software. On the other hand, there's the way which we like is you have something as a fixed annual expenditure where you pay 10, 20, 50,000, whatever it is for the software, and the software runs. It's as many times as you want to run the software, that's what you pay. The better thing about that, doing a licensed model, is that there's no need to pay for rerunning of samples, whereas if you have a sample run, you want to tweak some parameters, want to rerun it, the sequencing was bad, you have to rerun things, there's no need to pay for the software again. The other thing is we do a lot of research samples where it's very hard to get someone to pay for the software. Even things run on other panels, people just want to run a similar analysis. In research, it's hard to justify the software cost, and so in that case, also using a licensed model works better. So we selected for our analysis, going from the VCF file downstream, VarSeq, which is from Golden Helix. It's basically a graphical interface. I'll show some screenshots to filter variants and classify them using public and private data. It's roughly equivalent of taking Anavar or VATS and having a GUI wrapped around it. So anybody's familiar with SVS and what that does? For Plink, it's similar. Plink was one of the top softwares and still is for doing GWAS studies, where it takes all the GWAS data and produces results. If somebody is not a computer person, they could use Golden Helix, SVS, which is basically a wrapper around the software and does all the same things, but you have a GUI interface. And that's the same thing VarSeq did for this variant analysis. There's a command line version available, which we've looked into, where you can set up everything in the GUI, and then once it's set up in the GUI, you set it and it just runs automatically, whereas every time a sample BCF is produced, it goes through and the filtering is done right away. So it has a bit of a hybrid of the GUI and the command line version. The software is highly configurable, which is what we like, that you can, there's some finders we looked at where you have to have everything hard-coded, and if you want something changed, you have to call them and then they'll change it for you, but you can't just go yourself and create parameters. Here you can processing is very fast. It takes less than a minute per sample for our panel. If you have a bigger panel, such as a whole genome or whole exome, it might take a few minutes or a bit longer to process. Very quickly reduce the number of variants from a large number to hundreds to less than 10 variants. And as I mentioned, the software is purchased on annual license, which we really like. We pay the license, we have funding for it, and then we don't have to think about paying per sample after that. So the way the variant filtering works is that after you sequence, you get a VCF, you get about more than 100 variants per sample for our panel. This is for a small 50-gene panel. With exome or genome, you're going to be talking about tens to hundreds of thousands of variants. So everybody knows there's no easy way to go through those variants, and you have to prioritize and say which variants are most likely involved in the tumor and how can we deal with them. Most of the variants are not important. They can be germline variants, which are just that this person happens to have differences. Right? Every person has differences that are not pathogenic or not involved in the disease. They're just based on their ethnicity and their skin color, eye color, etc. So those variants we don't need to care about. Also, mutations that could be passenger mutations from highly mutating tumors, where you have mutations that are seen lots and lots of times in tumors, but they're known not to have any effect, so you want to filter those out. Also want to get rid of variants that have a low read depth, because the sequencers do have an error rate, especially when we're doing high-depth sequencing as we're doing. Even the low error rate creeps up to being a substantial number of reads, so you want to get rid of. The other important thing is looking at the minor allele frequency, because what happens is that you can't sequence just tumor. You're always going to have some stroma or surrounding cells that you're sequencing. And so when you find the mutation, you want to look at what your tumor content was and what the content of the tumor was for this mutation to see how likely the mutation is to be important. It's also important to find that a mutation that's very, very only found in a small percentage of the tumor is most likely not to be important. This different people do differently, but the idea is if the mutation is really critical and is giving the tumor a selective advantage, then it'll spread throughout the whole tumor, because a tumor is basically a very small evolutionary experiment where you're putting a cell in and these cells and what can make the cells grow and how can they help grow better. And so whatever mutations help it grow better, those would spread throughout the tumor and overtake the other cells. So for filtration, what we do is first make sure things pass the standard BaseSpace filters, which are things like strand bias, making sure the reads look good and the variant calls are good. Read depth we require is greater than 1000x. For New York State certification, you have to be greater than 500x, and so we pushed past that with this panel. Minor allele frequency we require more than 0.05, which we don't want things that are only occurring in a very small percentage of cells. That could be noise. We also filter things out. This is our main filter to get rid of things that are common variants. There are different databases. There's the exact, or there's the exome variant server. We use the filter at greater than 1% in any 1,000 genomes population. This was a data set that I think is compiled by Golden Helix, where anything that occurs in a germline in greater than 1% of people in any population is probably not involved in a tumor. And then we also get rid of things that are non-synonymous. The also thing which we get rid of, which is important for us, is we don't want anything that's a false positive in our assay. So every assay, every test is going to have consistent false positives, things that show up. So what we found is that when we do the sequencing, we always find the same mutation over and over in our samples. So once we did our first few dozen samples, realized these mutations occur everywhere, we just put them in as a bed file, put that file in, and then automatically gets filtered out by VarSeq. So here's a quick screenshot of how things work. So this sample had 182 variants, and it went down to 2 variants. So we're filtering passing the filters, allelic depth, minor allele frequency. Here we did 0.3. Sometimes we want to check different parameters, whether it's in this dbSNP common database, which is things that are greater than 1,000, greater than 1% in any 1,000 genome population. I can see what's ontology, which is filtering only things that are non-synonymous, and getting rid of the false positives. And you see here are the mutations, all the data about them. You see we went from 182 to 2 mutations very, very quickly. For the data sources, there's a whole bunch of data sources that are available free. You can just click and download them, and it'll be stored for availability. We've added in some of our own databases, where all you need is either a bed file or a VCF file. You throw it in, and then it just gets included in the software, which is very nice. And here are the results. You can export the results very easily as an Excel spreadsheet. This is spit out by the VarSeq software, and then they can be looked at by a clinician and then determine what to do. In our case, after we have the list, we have to determine what is relevant, what mutations do we need to deal with. Because even though something looks relevant there, we want to find out what mutations have our known drug targets, and what mutations could be a VOS or don't have a target yet, or maybe part of a clinical trial. And so to do this, to really understand the variants, you need to be a master of the literature. But the problem is for literature on variants, there are hundreds of papers coming out every week. Keeping track of all the papers is a very hard process to have someone read and classify all the papers. And on the same note, clinical trials are constantly opening and closing. And so setting up something to manage the clinical trials is very hard. Keep track of every clinical trial and what's going on with the patients, whether the clinical trial is allowing people or not. Because what happens is a lot of clinical trials will only accept patients who are sick but not too sick, have a certain set of mutations, and geographical location is important. So that's critical to do, and that's also requires maintaining a big database. So as an example of what's needed, this is from Ingenuity, another provider. You see they have people reading 300 journals every single issue. You have people reading, so you have a team of PhDs reading every single paper on every one of these journals and putting the results into a database. For further 3,600 journals, they do a combination of machine learning plus people reading papers. So it's a whole lot of work for people to do all this and to maintain this. We didn't have the resources to do this type of thing internally. So we turned to a provider called N-Of-One. So instead of needing to keep the database, we outsourced N-Of-One. N-Of-One worked for Foundation Medicine for a long time, providing their database, dealing with their data. And what the N-Of-One does is you're sharing in the idea of a database. They have a database of literature and clinical trials for the variants, and you just send them your variants, and then they process through the database and give you the result. The way it works is we export data from VarSeq as text or Excel file. We convert that to an XML file. The XML file is sent to N-Of-One. N-Of-One does that process of pinging into the database to see what the annotation is for those mutations. Sometimes the way it works with N-Of-One is some things are in their database. It's known mutation, something they've seen a lot that gets returned to us very quickly. Sometimes it's a mutation that requires a bit more work that they spend time on and can take a few days to get back to us, but overall it's a relatively quick turnaround. And then after the report comes back from N-Of-One, we send it to a molecular pathologist. He reads over the results and signs it. Rather than just taking the report from a company or from N-Of-One and just accepting it, we have a final level of review where a molecular pathologist reviews it, and he's signing the report, so it's a real standard clinical report. This report becomes part of the patient's electronic medical record and can be used to help determine the treatment for the patient. This is an example of what the report would look like. See for a patient what type of tumor it is, the specimens we got, where it's from, and the results you get. Here are the genes and the mutations, what pathway the mutation is in, the monocyte frequency, and then treatments that are available. So this is just a summary page. The doctor can look at this, see this very quickly, and then all the further pages go into details about what mutations. Here are VUSs, which are mutations that we're not sure if they're benign or pathogenic. That's a whole discussion about how to classify mutations in that category and what mutations matter or not. But this report is sent back to the physicians. And then the next step is the molecular tumor board, which we think is a very critical part of the treatment, is that you bring together bioinformaticians who look at the data, you have molecular pathologists, you have oncologists. Everybody discusses at a weekly meeting what they have seen in the samples, and then after this discussion they determine what the proper course of treatment is, taking into account not just the genetics but also patient's prior treatment, their condition, other comorbidities, and how to deal with it. And we think having all the professionals together does a really good job at getting things done and at helping treatment of the patient. The next step, which is critical for us, is the clinical data warehouse. As I mentioned, we're a research shop where we really want to work on these patients. So we take the molecular test results, along with any diagnostic and treatment data from the EMR, put them into this big database that's queryable. So now, with appropriate IRB and HIPAA controls for the data, you can query and ask a lot of innovative research questions. So you could say, like, let's take breast cancer patients that had a certain mutation, how did they do on a drug? You could do a retrospective trial very well by just querying this data, and you can also look at people and say maybe people who had certain other sample characteristics from their blood or from their histology, how does it work together? Next level is you could also look at, so mutation A is indicated in helping determine if someone should get a drug, but does it really mean that instead of one mutation, I use this mutation plus 2 or 3 other mutations, gives me a better prediction of how well someone will do on the drug, and that's doable. So this is all put together in the clinical data warehouse, and we find that a very powerful resource for either just looking back at our patients or designing future trials. So just a quick summary. Clinical tumor sequencing is directly changing cancer treatment rather than just treating patients with a tumor as having cancer and throwing chemo at them. We have targeted therapies. There are a large number of computational steps to do this and web lab steps using Raindance, VarSeq and N-Of-One to do this analysis. But through these panels, we've enhanced the outcomes for our patients, and we find it to be very promising as a way to treat our patients. Thank you very much for your attention. I'd be happy to answer some questions if people have any.
Thank you so much, Jeff. Pardon me. If you do have questions, feel free to enter them into the questions pane now. I am recording today's webcast. I did see that question out there, and I will email it to everyone just as soon as it's ready to go. That should be on Friday at the very latest. Our next webcast will be coming up on the 21st, and I am working on the invites now, and you should see those tomorrow. There's lots of thank yous in the questions pane, Jeff. One question here, does your report content come from N-Of-One? I think that's how I'm reading it.
Yes, the content mainly comes from N-Of-One. We do some spot checking or look over some papers. If something that's novel, we might check it, but it's mainly reliant on N-Of-One.
What proportion of patients require further sequencing beyond the 50-gene panel?
It's a very good question. I don't have enough data really to say that as of now, but it depends on the patient and the tumor type whether we can find a variant easily or not.
Is the N-Of-One service pay per variant?
So N-Of-One, I think their pricing depends on the institution. They have different ways of paying per sample, paying per variant. I know we have a certain plan, but I don't know whether that's just for Rutgers or how that works for different institutions, so I don't really want to get into N-Of-One's pricing, but it's definitely either pay per sample or pay per variant thing. They do not have an annual license version.
What is your opinion on whole genome versus exome versus gene panels? At $1,000 per patient, whole genome sequencing seems like the way to go.
So the $1,000 whole genome you're talking about, which is what you can get from putting on the Illumina 10x, that's a 30x genome. So what we need for this is at a minimum 500x or ideally 1,000x. So you're talking about way more than $1,000 for your genome just for the sequencing. And so that's not feasible. If you just want to do a low coverage genome for something that's not really important, where you're not really caring about every mutation and in a tumor, then that $1,000 works. But for a bigger panel, you can't justify doing whole genome as of yet.
Oh. Do you have numbers on how this pipeline is improving the success of the used treatments?
I don't have data. We had a paper that came out recently going through our molecular tumor board and showing that for most people, there is a therapy that we find. Whether they can go on the therapy is dependent on a lot of the clinical characteristics, how sick they are, what other comorbidities, and how things are working. But this is definitely improving treatment.
Which source do you use for determining off-label drugs for potential cancer treatment?
So the source of this is all we're getting from N-Of-One. And you just look at the database and you see that FDA approval is for a certain mutation in a certain type of tumor. And so if you have that mutation in a different type of tumor, then that's an easy way to say it's off-label. The publication is discussing whether this is really a good idea or not, or whether people have tried some off-label things. Some off-label work, some don't, and so that's all dependent on just publications.
Sorry, bear with me here just a minute. They're popping in so quick. If the database fed-in software doesn't contain population-specific allele frequencies, how accurate is a variant call as an actionable mutation?
If what database doesn't? Say that again.
If the database fed-in software doesn't contain population-specific allele frequency.
Okay, so the data from 1,000 genomes and dbSNP, you can get data on populations. So based on that, you can look at, and so you're right, we don't know the specific population of this mutation. You could have cases where where a mutation occurs in one population and is pathogenic not in another population. I've thought of that for a long time. It's if it really occurs, it's very, very rare, very few cases. So any mutation that occurs in any population at a significant percentage is very unlikely to be pathogenic.
Why filter out mutations greater or equal to 1% in 1,000 genomes? Also, oh, I'm sorry, there's a second portion. Also, why passenger mutations are not desired? Looks like someone missed your answer.
The passenger mutations are not going to be involved in the treatment because a passenger mutation is just a mutation that is not involved in the treatment. It's just something that happens from tumor being highly mutable, and so that's not going to be important. And as far as the 1%, we just want to have a very strict filter to get rid of things that are likely to be false positives. These things that are tumor mutations are very, very unlikely to be found at all in germline, and so that's why we have a harsh filter.
Alrighty. We are going to go ahead and wrap today. I will make sure that we get the rest of these questions answered. You can look for those via email. Jeff, I appreciate your time today, and I appreciate everyone's attendance, and we will see you next time.
Okay, thank you very much.
Have a great day, everyone.